See a double helix? Put your right thumb up. If your finger's tracing, the graphic designer is in luck. So DNA is present as a right-handed double helix, and when you can check if a helix is right-handed by curving your hand, and your finger should trace out the curve of the DNA. And so in the DNA, there are two strands, so it's anti-parallel. So one strand is running in one direction and one strand is running in the other direction. The unique parts of the DNA, their bases, are so like the A, T, G, all that stuff. Those where they form these complementary base pairs with each other. We'll talk about A to T and G to C. These are going to be in the center of the helix, and on the outside are these negatively charged phosphate groups. So you have the sugar phosphate backbone. But they're not just like straight parallel like this. Instead, they curve around in this double helix. And so the double helix is because it has this big major groove and this little minor groove. And there's different information that's like proteins, like transcription factors, so things that need to access the DNA in order to like make messenger RNA copies of it to make protein from it. We'll get into all of this. But you can get different information. They're kind of like windows into what's going on inside of the DNA in those bases, and you can learn different things about them from these different grooves. Also, there's more space to interact with the major group, so you can figure out more there and you can access it more easily. So today I want to tell you more about these various aspects of the DNA structure, and how we can think about DNA structure as everything from the individual letters themselves, so from those nucleotides, all the way to these double helixes. And the double helixes is just like the beginning, because then they wind up, they coil up, because you're so much DNA. Like if it were just in that like single double helix, like really, really long, like super duper long, you would never be able to fit it in a cell. But instead it coils up really tightly with the help of these proteins called histones, and so you can get like modifications to those histones to help make regions more or less accessible. And this is a region we call like epigenetics. So there's all these different layers of DNA structure, and we can understand all of what's going on better if we understand the biochemistry of DNA. And so let's get into it. So just an overview, and then we'll dive in. So we can think of DNA as having lots of layers of structure, similarly to how we could think about proteins as having lots of layers of structure. But with DNA, DNA is kind of like simpler in terms of you only have like 4 different letters instead of like 20 like you have for proteins. And the structures that you're going to get are basically all the same. And so these are going to form this double-stranded helix where you have these unique parts of these nucleotide letters are going to be hidden in the center of this helix. And then this helix is actually going to get really, really tightly coiled up. And so although we often think about seeing like DNA as this double helix, this helix itself is going to be tightly coiled up around these proteins called histones, and so that they can get stored in this nucleus. And we'll talk later about how we can modify these histone proteins and do things with like epigenetics in order to help make regions more accessible when you want to do things like make messenger RNA to make a protein from them. So there are 4 DNA letters and 4 RNA letters that we'll get into, and these letters are called nucleotides. These nucleotides all have this like generic sugar phosphate part. And then they have one of 4 different bases, one of 4 different nucleobases. How these bases are different, well, you can see that some of them are big. These like purines have these 2 rings, and the pyrimidines have a single ring, but they also can form like base specific pairs to one another through these like hydrogen bonds that we'll talk about. But you're always going to have one of these big guys and one of these little guys, and so your helix is going to have this even width. When you have this helix, you're going to have these bases are going to be on the inside, and the phosphate chain is going to be on the outside. The base pairs are really important for making specificity and for allowing you to use one strand as a template for making the other. But the real effect of helping keeping this DNA really in this like structure and keeping it all stable and tightly bound together is actually happening through these things called base stacking, which are happening like between these different, between the bases that are stacked on top of each other, and they're stacked kind of staggered. And so because they're like kind of offset, this is going to make your DNA into a helix.
And you have these DNA bases. They have this quality, they're called like hydrophobic. We'll get much more into all of this, but basically they're excluded by the water. And so they're going to be in the center of this helix, whereas these hydrophilic phosphate groups, so these are negatively charged and water really likes them, so they're going to hang out on the outside. And then the end result is that you're going to get this double helix where then you have this major groove and this minor groove, and there's different information that different proteins can tell based on what they see if they will look through those windows. So now that we've gotten into the overview, let's get into a little more detail. So DNA is the way that our bodies store permanent genetic information, including like the original recipes for making proteins. So we store our DNA tightly wrapped up in this part of the cell called the nucleus, this membrane-bound compartment. And then when our cells want to make proteins, we first make RNA copies of them that get shipped out into the cytoplasm, so the general cellular interior, and then can be used to make protein. So all of this whole situation requires that, one, our DNA be really stable, two, we're able to make RNA copies of it, and then that those RNA copies can be used to make protein. And so we've talked about how we can use the RNA to make protein in the process of translation, which we talked about a lot recently. But today I want to focus on the DNA. So proteins and nucleic acids are both biopolymers. So a polymer is just like a chain of similar subunits. And so we call those individual like subunits monomers. And so with a protein, our monomers would be amino acids, which have these generic backbone with this amino group and a carboxyl group that link up through peptide bonds, and then these unique side chains that stick out. With nucleic acids, which include DNA and RNA, our monomers are going to be nucleotides, and they have generic and unique parts as well. The generic part of these are going to be the sugar phosphate backbone, which has a ribose sugar in RNA and a deoxyribose in DNA. So it's missing this OH group on this. But then they link up, instead of through peptide bonds, they're linking up through phosphodiester bonds, which are using these phosphate groups. So with amino acids, we had like all those 20 different side chains, and they were interacting with all different things in really unpredictable ways, and in a lot of different ways. When we have DNA and RNA, the really core part of the uniqueness of them is that they will specifically base pair with one other base. So when you have the DNA strands, so DNA likes to exist in this double-stranded manner we'll get into. And so those bases are going to be facing each other. And the binding opportunities for these things called hydrogen bonds or H bonds, they're going to be optimal when you have a G and a C, and when you have an A and a T, or an A and a U. And so because you have the specificness of the base pairing, then you can use one of these as a template for one strand as a template for making the other strand. And this makes the DNA a really good source of storing genetic information. So we can use enzymes, so like reactions helpers, like reaction like mediators, speeder uppers. These proteins can help us go from like DNA to DNA, DNA to RNA, et cetera, et cetera. In order to do that, of course, they're going to have to pull apart the strands of the DNA. So let's look in more detail about how the DNA structure is actually formed. So first, let's get some terminology out of the way. So with DNA and RNA, so with both of these, you're going to have a nucleobase or a nitrogenous base, plus a pentose, so a 5-carbon sugar. When you add these together, you get a nucleoside. So nucleobase plus the sugar is a nucleoside. So you can remember S side, S sugar. So when you add a sugar to a nucleobase, you get a nucleoside. Then if you add the phosphates, then you get a nucleotide. So phosphate, nucleotide. So that's how I remember. The T goes with nucleotide for the phosphate, and nucleoside when you add the sugar. Things get kind of confusing though, in that when you add the phosphates, when we name them, so we can add different numbers of phosphates. So we can get, if we add 1, we get like a monophosphate, 2, we get a diphosphate, 3, we get a triphosphate. But we're going to refer to them as a nucleoside triphosphate because it would be redundant if we said like the nucleotide triphosphate. So these nucleobases, there are 2 basic forms. You have purines, which are these bigger ones, and then pyrimidines, which are these smaller ones. So the purines have these 2 rings, the pyrimidines have this single ring. And how I remember this is like purines, like they're pure, they have it all. And so they have like 2 rings, and then pyrimidines is the other thing. So basically, I think of the purines as like having it all, and then the pyrimidines would have like lost something or something. That's just a memory trick that I use. But there are different ways, whatever helps you remember that the purines are going to be the bigger ones that have these 2 rings, and the pyrimidines are going to have this 1 ring. So there are purines. So another memory trick you can use to remember which are which is that pure as gold. So purines are adenine and guanine, and then the others will be your pyrimidines. So cytosine, thymine, and uracil are the pyrimidines. When we go from a nucleobase to a nucleoside, so when we add a sugar, the names change. So if you have, when you have the purines, they're going to get this ocene ending. So adenine becomes adenosine, guanine becomes guanosine. Cytosine, like this thing just like trips me up because it has seen in its nucleobase name, and then it becomes cytidine. And then thymine is going to become deoxythymidine, and uracil is uridine. So if you have a pyrimidine, it's going to become get the dine added to the end. So pyrimidine is going to give you a dine something, and then adenine and guanine, so your purines, are going to give you the ocenes. And then if you add the phosphates, then you add the like monophosphate, diphosphate, triphosphate, etc. So when we talk about like where things are attaching, when we're talking about, when we number the sugars, we use this like prime nomenclature. So primes is like we have a little like dash mark. And so this is going to be your 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So this is your 5 prime OH, which is where the phosphate's going to get added. So we can say that you're going to have the phosphates are attached to the 5 prime. 1, 2, 3, you have a 3 prime OH, which is where the bases are going to join together. And then with RNA, you have ribose, which has this 2 prime OH as well, whereas in deoxyribose, you don't have that OH group there. So RNA and DNA differ in that RNA has this 2 prime hydroxyl group that DNA doesn't, and they also differ in that RNA has uracil and DNA has thymine instead. But uracil and thymine can both pair with A, and this is why you can make like DNA from RNA and RNA from DNA in terms of like using one as a template for making the other. RNA and DNA also differ, and so having this OH makes the RNA less stable. The RNA also tends to form more diverse structures and not, so DNA, you're pretty much always going to find it in like the double-stranded form, but RNA often find it single-stranded with like loopy regions like bonding to itself and that sort of thing. The phosphate groups of the incoming nucleoside triphosphates, they're going to provide the energy that's needed to help make it favorable to link these up. So when we're talking about amino acids, we were talking about how we had to go through this like convoluted pathway of charging the amino acids and the tRNA, all of the stuff to get the energy to go. When you're talking about piecing together these nucleotides, they have the phosphates already. You attack the 3 prime OH is going to attack the 5 prime phosphate. This is going to kind of provide the energy needed to get this reaction to be favorable. And this is done with the help of a polymerase. The consequences of having it occur in this 5 prime to 3 prime manner is that at the 5 prime end, you're going to have a phosphate group, and at the free phosphate group, and at the 3 prime end, you're going to have a free OH. And by convention, we write DNA from the 5 prime to the 3 prime. So if you see a DNA sequence, it's probably written 5' to 3'. And then on the other strand, so it's base paired to its reverse complement. And so basically it's the reverse complement because this strand, we're numbering 5' to 3', this strand is anti-parallel. So it's base pairing to the one that it would normally base pair, but the strands are anti-parallel. So if you wanted to write this strand from 5' to 3', then you need to add, you would write the reverse complement. So the terminology can get kind of confusing, but the basis for this base-to-base specificity is the base pairing. So when you have the base pairing, you're always going to have a purine and a pyrimidine. So you're going to have one of the big ones and one of the small ones. So before we even really knew that much about the DNA and all of this, there was this Shargrass rules. So the scientist Shargrass, he found that the ratio of purines to pyrimidines in like all of these different types of cells and all these different organisms was like 1-to-1. So it seemed that there was an equal amount of purines and pyrimidines, and this can be explained because you have one purine and one pyrimidine in a base pair. More specifically, you're going to have a G to a C, an A to a T, or an A to a U. And because you have this, one of the big ones and one of the small ones, you're always going to have an even width in the DNA helix. But where are these bonds coming from? So these are what we call hydrogen bonding. The basic idea is that they're like partial charge attractions. And so all of these atoms that we're looking at, they're joined together by sharing these negatively charged parts called electrons. And so these atoms have these like electron clouds, and when they form these strong covalent bonds, like the bonds that are actually like holding together like the individual strands, like holding together all of the individual bases, not like between strands, but just like holding the molecule together so it's not just like disintegrating. So all of those strong covalent bonds, those form by like merging their parts of their electron clouds kind of. If the atoms don't share fairly, then you can get an uneven distribution of the charge. So the atoms, they have all the atoms have like positively charged parts called protons, neutral parts called neutrons, and then these negatively charged parts called electrons. The protons are stuck in place. So you can think of kind of like the protons as being in the center of this O, the center of this H, and the center of this H. And they have a fixed number of these protons, but then the electrons electrons have an equal but opposite charge. So if you have an equal number of protons and electrons, you're going to have an equal a neutral molecule, such as with water. But with water, it has this thing called polarity. So basically the oxygen is going to hog, it's what we call electronegative. It's going to hog the electrons that they're sharing. This is going to make the oxygen partly negative and the hydrogen partly positive. So the electrons in those merged clouds is like hanging out more with the oxygen because these electrons are really whizzing all around and we can't even predict like exactly where they'll be, but we can say, oh, they tend to hang out over here more. If they're hanging out over here more, this part's going to be partly negative and this part's going to be partly positive. When you have opposite charges, they're going to attract. And this is even true of partial charges. And so we can get, if we have an electronegative atom, one of those hoggers that has like a lone pair of electrons, so an unbonded pair. And then we have a hydrogen attached to an electronegative atom. So hydrogen is basically really bad at holding onto its electrons. It only has like a single proton to try to like rein its electrons in with. So it's not very electronegative. And so if it's attached to something that is electronegative, this is going to make the hydrogen partly positive. Then you're going to have it, you can have it interact with that lone pair, and this thing we call a hydrogen bond. So we call it a bond, but it's really just a strong partial charge attraction. And it has another thing about hydrogen bonding is it likes to have this this like linear relationship between like the lone pair, the the donor and the hydrogen. And so this is going to help be the strongest when they're like this, and this helps explain some of the structural concepts. So if we look at the bases, we can see with adenine and thymine, we have two so with adenine and thymine, for each of these, we have one donor and one acceptor. So our donor is hydrogen on the nitrogen, and then our acceptor in adenine is nitrogen, and thymine is an oxygen, the lone pairs on them. And so these, when they're running anti-parallel, they can form these hydrogen bonds with one another. With guanine and cytosine, here we have the opportunity to form 3 hydrogen bonds. And so you might think, okay, well, this explains why you might have heard that CG bonds were stronger than AT bonds. So you can see that with GC bonds, you have these 3 bonds, and with A and T, you only have 2. So this is true in terms of like why these individual base pairs, if they were on their own, would be stronger. But this isn't the reason why GC-rich regions of DNA are like harder to melt, so harder to pull apart. Instead, it has to do with base stacking. So we've been talking about hydrogen bonding, but we've been ignoring the one of the major forces in actually holding the DNA, giving the DNA stability, which is this thing called pi stacking or base stacking. So when we talk about atoms as like sharing electrons, so normally if you have just like a normal single bond, it's like sharing a single pair of electrons. A double bond would be sharing 2 pairs of electrons. And then we talked about like lone pairs, which are unbonded. You can also have this thing called resonance, and then where the electrons are kind of like delocalized. And then this happens in a ring. We call these aromatic. And so we can talk about having these like pi bonds, where pi bonds. So and in the case of a pi bond, it could be like just like a normal double bond. But in the case where you have delocalized pi bonds, this is where you have it like delocalized, and if it's in a ring, we call it aromatic. And so it involves this stuff that's too complicated to get into here, but it involves these like p orbitals, which are located like above and below the plane of the ring. So we have this, so when we have like these pi bonds and aromaticity in these rings, what can happen is that you have all this electron density above and below these planes. So we talked about like partial charges attracting. This is true even if those charges are just like temporary. And so what can happen is we saw in the case of like an electronegative with like polarity, how we had an electronegative atom kind of like pulling the electrons and making a region partly negative and a region partly positive. But because those electrons are kind of just like whizzing all around, this can happen like randomly, that the electrons just like happen to be in one area more than another, or they can become like polarized if something charged come by. So you can imagine if you have something that's like charged or partially charged, then it can be if you have something like negatively charged come, then the electrons that were hanging out over here on this side of the molecule, they're going to get repelled and then hang out more on the other part of the molecule. Then that's going to cause like a kind of chain reaction. And what's going to happen is that you can get these all kind of like in sync in terms of their electron clouds, and this can provide great strength. So when we look at like all of these bases stacked together, if they're all like syncing up their clouds, you have this kind of like all these stacked on stacked on stacked. You can think of it as providing a lot of stability, that you're having all of these interactions, these pi stacking interactions. And it turns out that these pi stacking interactions are sequence dependent. And so when you have like GC sequences, you're going to have a lot more, it's going to offer a lot more in terms of stabilizing effect. And so I found this really nice video by Derek McLaughlin I recommend, and it goes into, so from this study what they were measuring the delta G of stacking. So basically, what's the energetic benefit of having a stacking interaction, or like how costly would it be to break it? So basically, the more negative this is, the better, the more benefit you get. So if you have so you can see with like a GC-GC pair, you're going to have the strongest effect, whereas with like an with a TA-TA pair, you're going to have a weaker effect. And so you can see that this is all sequence dependent, but this is the basis for the main basis for why GC-rich regions of DNA are stronger, why you need to they have like a higher melting temperature. So basically when you're doing PCR and you're trying to, the amount of the heat that you need to add, so the energy you need to add to actually like pull these strands apart is going to be greater if your sequence has a lot of G's and C's. This is not because they have 3 pairing instead of 2 pairing, but the main reason is really because of the differences in their base stacking. But the hydrogen bonding, of course, is really important for determining the specificity, the base pairing specificity. So what binds with what? It's just not the major driver for stabilizing the DNA helix. So that kind of like polarization of those pi bonds and these various interactions, we call these like van der Waals interactions, where you just kind of have those like random random kind of charges and then like polarize other charges, and you kind of just have things sync up. So instead of having like fully some molecules are basically like permanently polar, that sort of thing, or permanently charged. So these van der Waals attractions and stuff can happen even in a molecule that is normally non-polar. So in a molecule where all of the atoms are sharing fairly, or at least fairly. And so this is the case when it's like hydrogen and carbons. So when you have like hydrocarbons. And so when we look at these DNA bases, we can see that there's a lot of like these hydrogens and carbons, and we have a lot of this hydrocarbon character, even though there are some like heteroatoms, so some like nitrogens and oxygens and stuff. There's a lot of this non-polarness. And this has the effect of making those bases hydrophobic. So basically, there's this thing called the hydrophobic effect. So because water has that polarity, it has a strong permanent polarity, where the oxygens are partly negative and the hydrogens are partly positive. And because water can like hydrogen bond with itself, water is going to want to like form networks with other water molecules. And it's only going to want to like let in other stuff if those things have like partial charge or full charge, because it doesn't want to give up its water-water interactions in order to like accommodate something that has nothing to give it. And so this is why, for example, like oil, which is really non-polar, isn't going to mix with your water. It's not going to dissolve in water because water wouldn't want to. It doesn't offer the water anything in return. And so we say that things that water will let into its network are hydrophilic. So these include things that are polar and things that are charged, that sort of thing. But things that don't offer water anything, so things that are like non-polar, are going to be hydrophobic. And we can talk about different regions of the same molecule being more hydrophobic or more hydrophilic. So if we look at DNA, we can see that this sugar phosphate backbone, well, that's going to be really hydrophilic because it has this full charge. And so water likes the partial charge of other water, but it really likes the full charge of these phosphate groups. And so this part is going to be hydrophilic. And so water is going to be happy hanging out with it. But the bases, they have a lot of hydrophobic character. So they have this these parts are more hydrophilic, so like the insides that are actually, but these are going to be paired to one another. And then you have this hydrophobic part. And so if this hydrophobic part were like sticking on the outside of the double helix, then it would have a lot of area that the water would have to like give up water interactions to hang out with. So typically, if you put something hydrophobic into water, water is going to try to minimize its interactions with that thing and maximize its interactions with other waters. This is going to kind of push the hydrophobic things together. This is going, we call this like the hydrophobic effect, and it's going to be a main driving force in getting the DNA strands to get into this like double-stranded helix form, because you're going to want to kind of like, the DNA is going to want to exclude the hydrophobic parts of the bases, push them into this center. And when they're in the center, then you can get those like big networks of these like pi bonds in these pi stacking and that sort of thing, as well as having this hydrogen bonding. So you have the water exclude, like the hydrophobic effect kind of helping facilitate and strengthen this bonding process, where you have the nonpolar parts kind of hidden and in the center of the helix, and kind of offering each other interactions that they can't get from the water and that the water doesn't want to get from them. But the basic idea is that in order to maximize the base stacking interactions, you don't want to be like directly on top of each other. You want to be like slightly offset. This slight offset is going to make it so that the DNA is going to like be in this helical form, or at least that's my understanding. Like there are other forces that work as well, including things like the sugar puckering and the base pairing. But having the bases at that offset is going to give you the maximize those van der Waals interactions, maximize those that like pi stacking, and have the consequence of giving you this major and the minor groove. We know about all of this thanks to photo 51, which is a fiber diffraction image taken by Raymond Gosling, who was a graduate student in Rosalind Franklin's lab, and the work that they did figuring out the various, finding this structural picture that helped Watson and Crick make the needed calculations and publish the results. So some proteins can like read out genetic information without pulling the strands apart, which is great because you don't want to have to pull those strands apart if you don't need to. If you were a protein, you would have trying to read out some information, you'd have an easier time accessing the major groove, which is great because the major groove is actually where you're going to get the most information. So you can think of these grooves as kind of being windows into what's going on inside of here. If you are looking from the major groove, you'd be able to tell apart every base pair. So you can tell apart a GC versus a CG, in addition to be able to tell apart an AT versus a TA. So you can tell all four of these apart. So if you're in the major groove, so there are four basic things that you could see if you looked in one of these grooves. And so you can see a hydrogen bond acceptor, which I'm showing as an A, hydrogen bond donor, which I'm showing as a D, a hydrogen itself, which I'm showing as an H, and a methyl, which I'm showing as an M. So with a donor, it'd be a hydrogen that's attached to an electronegative atom, and with just a plain hydrogen, it's just like attached to a carbon. So it can't participate in hydrogen bond donor. But if you were to look at like GC, you would see AADH. If you were looked at CG, you see HDAA. So you can tell that these are different. If you were to look at AT versus TA, you'd see ADAM or MADA. And those are both different from this. So if you were to look, you would be able to tell this base pair apart. So and remember that GC is not the same as CG because these are on different strands. So you could have a G on this strand and a C on this strand, or a C on this strand and a G on this strand. So those are different. But if you were to look from the minor groove, well, now you can tell that a GC, that something has a G and a C versus having an A and a T, but you can't tell the order of them. So you can't tell GC versus a CG. In both cases, you see ADA. And in AT versus TA, you would see AHA. And so you're getting more information from the major groove view, and it's easier to access. So I don't have a memory helper for this one, but I came up with this memory helper for AT versus TA in undergrad. So basically, atom H automata. So if you have, so AHA, you're going to see with either A or T. Atom 8, so when you have an AT, you're going to see ADAM. a tomata. So when you have a TA, you're going to see MADA. So that's just my weird little memory helper if that helps you. So hydrogen bonding can also occur like between the bases and water. But when you have it in the DNA, when you have it between strands, you kind of have this like cooperativity effect where basically if you have one like next door or whatever, it's kind of, you're already there. You can just like bind right away. And also having them like in the center away from the water. So when you're in this double-stranded manner, then it's going to help give you those like base band bonds instead of like base water bonds. The phosphate backbone, so in addition to forming like bonds to the water, it'll also attract like counter ions, so like positively charged metal cations, things like magnesium. So typically what happens is you have a water layer kind of helping mediate between the two. So these phosphates are going to have this layer of water, which is going to then like through like polarization, like through the water, like you kind of have your charges line up. And so then there's going to be like a cation, like a magnesium and stuff helping here. Because this DNA is normally all like coiled up and stuff, and in order to make a protein from the instructions, we need to basically make an RNA copy of the DNA. So if we want, we can regulate how many RNA copies we make by regulating transcription. And so this is going to involve like transcription factor proteins binding, etc., and how accessible the region is. So basically you have the DNA all wrapped up in those histones. When you have it like tightly coiled, we call this like heterochromatin. And when it's like looser, we call it like euchromatin. But basically you can have these histones, they can be modified and they can be like slid and stuff, or totally removed in order to kind of loosen up regions that you want the protein to have access to. The RNA polymerase, which is going to make the RNA copy of it. And then the RNA polymerase can get helped finding the site by like transcription factors, which are going to bind to like promoter sequence in front of the genes to recruit the RNA polymerase and help get things going. And the chromatin can be modified in ways that both like make it looser or less loose, and can also provide like binding sites for transcription factors, etc. And these histone proteins can be modified typically through like lysines and their tails. And so these histone proteins have these long tails with a bunch of lysine, the amino acid lysine. And as we talked about more in the lysine post, basically these can get methylated or they can get like acetylated. And so normally these lysines are positively charged, and so they're going to be kind of like helping neutralize the negative charged DNA and help keeping tight this like these are positively charged, and they're going to be like attracted to the negative charge of the DNA and help keep things kind of like really tightly wrapped. If you acetylate it, then you make it neutral, and so acetylation tends to be associated with a like opening up with increased transcription of a region. Methylation, the effects are going to vary based on where it occurs, what binds it, etc. And so these we call these like epigenetic modifications because they're like above the genetic level. So with the genetic level, we're talking about like what the actual sequence is. And then with epigenetics, we're talking about like modifications that we make above on top of that like genetic information that's encoded in the sequence. So I hope that helped you get a better appreciation of DNA at all of its structural levels.